Hi, everyone. This is our presentation for machine learning. The title of our project is called Predicting the Classification of Proteins in the AAA plus protein superfamily. And I'm just going to give us a quick background to start off with. So essentially, proteins are polymers that exist in all living things. These proteins fold to form three-dimensional shapes that help catalyze and allow certain reactions to happen. A big question in biology is how certain proteins classify into different families. And so one protein will say be in a family of DNA polymerases, and another protein will be in a family of, say, chaperones. And these proteins can be represented as linear string sequences as shown here, so this MVLS sequence. So the idea is to use this linear sequence without any other information to predict which proteins go into which families together with each other. So for this protein family AAA plus, we decided to figure out whether or not we could sort proteins in or out of this family. The data that was used was manually curated. So AAA plus protein sequences were manually found through literature search, and non-AAA plus proteins were mined from the UniProt database to design both testing and training data. And I'll hand it over to Andrew.
Our methodology included both an unsupervised set of algorithms and a supervised set of learning algorithms. The precursor to either of those items is the three-mer parsing algorithm, in which you take a sequence and parse it into a sequence of three-mers. And you would actually get, for each of the sequences, three separate catalogs of three-mer sequencing as shown in the step 1 and step 3. Following that, we first pass those three-mers into the unsupervised word-to-vec training model to essentially get the trained embedding, which will be necessary to translate our AAA positive and AAA negative protein sequences for the supervised section. Going forward in step 4, we take the training set that we would like to use in our logistic classifier, gain the embeddings for the input for the logistic classifier, place a positive for the AAA positives and a negative for the AAA negatives, send it into a regular logistic classifier to train that classifier, then finally take our protein of interest to pass through the classifier to get either a yes or no judgment on whether or not the protein of interest is or has the AAA features. I'll pass it on to Matt.
Great. So as you can read in our paper, we have a couple of hyperparameters, which are the number of positive examples to feed in, the number of negative examples to feed in, as well as the same for the test examples. And then the number of features, the context size, and then the minimum word count. Context size is how many words on each side of our three-mers to consider for the algorithm. And the features is how many, what dimensionality to use for the algorithm. Our confusion matrix, you can see in the paper, but our results were quite satisfactory on the whole. In the bottom left-hand corner there on the results, you can see that we got 99% on the training set and 96% on the test set for precision, and 96.3 for the training set and 92 for recall. And this is great because it's obviously an economical way to distinguish these AAA proteins from a random set. And I think that's all I have. Throw it back to Yusuf for any concluding remarks.
And so in summary, we developed a machine learning algorithm to classify whether or not proteins were in the AAA+ family or not. Future goals include whether or not we could apply this to larger families and use a larger training set to get better ROC curves. Thank you.